Hi, we are the Mightychondrias Unknown Hormone Group, code 574. Our members are Carissa Martin, Gogo Karangwa, and Meg Rossolo. Let's get started. First, we'll start with our objective to identify which hormone was administered to our rats. In this study, we were given adolescent rats that were approximately 5 weeks old and weighed about 100 grams at the start of the study. All of the rats were injected with the unknown hormone for 10 days, and the control group of rats were only injected with saline. The hormones that were administered are very high doses and frequencies during that period to ensure the changes were significant enough to be noticed. They received a dose from 1 to 4 times a day. We started off by reading the information on the project and all the different tests that we could order. Our second step was to compare the control data to our experimental data. We had code 574. We studied the list of hormones and used our information to carefully use the process of elimination. Which hormone is it? There was a possibility of 10 hormones that were administered to our rats. TSH, which promotes the growth of the thyroid gland in the neck and stimulates it to produce more thyroid hormones. Thyroxine, the main hormone produced by the thyroid gland, acting to increase metabolic rate and so regulating growth and development. A glucocorticoid, any group of corticosteroids which are involved in the metabolism of carbohydrates, proteins, and fats, and have anti-inflammatory activity. There was ACTH, a protein hormone of the anterior lobe of the pituitary gland that stimulates the adrenal cortex. Growth hormone stimulates the growth of essentially all tissues in the body, including the bone. HCG, a hormone secreted during pregnancy by the placenta, which stimulates continued production of progesterone of the ovaries. Testosterone hormone, made mainly in the testes, needed to develop and maintain facial hair, deep voice, and muscle growth. Estradiol, estrogenic hormone secreted by the ovaries, aids with maturation and release of the egg, as well as the thickening of the uterus lining to allow a fertilized egg to implant. Epinephrine, hormone secreted by the medulla of the adrenals, which causes an increase in heart rate, muscle strength, blood pressure, and sugar metabolism. Placebo, which is a substance that has no therapeutic effect. This is the control data and the experimental data for the rats in our project. We graphed our data to point out the significantly different data and the data of interest. We discovered that the following were significantly different: the thymus, the ovaries, the uterus, the seminal vesicles, and the ventricle prostate. Of interest, we found that the tibia width and the pituitary were of interest. All the others were no change. That included the body weight, adrenals, thyroid, of how large and the weight, the testes, the plasma, and the glucose tolerance test. Our glucose tolerance test results showed that even though the glucose in our rats was elevated more, it was still controlled. Therefore, we had no significant difference. We eliminated thyroxine, growth hormone, placebo, epinephrine, a glucocorticoid, TSH, and ACTH. Our possibilities of our hormones based on our testing was testosterone, estradiol, HCG. The first test we ordered was an RIA test for testosterone levels of both sets of rats. These results weren't as expected because the data we interpreted, everything pointed to testosterone, but the levels were close. If there were testosterone to be injected into the rats, the levels based on the experimental rats would be a lot less, but however, we see here that it is very similar to the control group.
Our second test we requested were complete vital signs for the control and experimental rats. The blood pressures and temperature of the control rats were significantly different. The almighty lab assistants so graciously passed on their behaviors with their vital signs. This helped us pick between which hormone to request the RIA of. with the increased mammary glands and the defensive behavior led us to pick estradiol. We decided to use our third and final test to see the RIA results of estradiol because the difference in the vital signs between the two groups. After our third and final test, we were able to eliminate HCG and choose our hormone. We found the results from our test that the rats were injected with estradiol. Here are the website that we used to distinguish between the hormones that we thought were injected with our rats. That is the end of our PowerPoint, and thank you for your time.